Hi, everyone, and welcome to our discussion on plant reproduction and plant structure. Before we hop in, I just want to say that this lecture repeats and builds on some of the core concepts discussed in our prior presentation on seed plants. As the presentation on seed plants provides an excellent base and launching pad into this conversation here, so I recommend you go watch that video or review your notes from that last video. In the last chapter on seed plants, we saw that there were many adaptations that emerged in seedless plants first that were necessary for the transition to land, such as the development of embryos, lignin, roots, and vascular tissues. However, one major limitation still remained, and that was the requirement for a watery environment in reproduction, both because we needed water in order to allow fertilization to occur, and also because embryos were not as well protected in the seedless plants as they were in the seed plants from desiccation. The evolution of seeds and pollen allowed new varieties of plants that possessed this adaptation to spread far and wide across land. Seeds and pollen could travel farther distances, carried by different mechanisms, and spread to new habitats. To understand plant reproduction, you'll need to have a grasp of their unique sexual cycle. We showed in the last presentation that all land plants, seedless and seed bearing alike, display alternation of generations. This life cycle alternates between these two multicellular forms called the diploid sporophyte phase and the haploid gametophyte phase. The diploid sporophyte is aptly named because it produces haploid spores through a process called meiosis, and these spores are released and divide by mitosis to produce a multicellular haploid we call the gametophyte. The gametophyte is named for the fact that it then produces haploid gametes, eggs and sperm, again through mitosis. When a sperm fertilizes an egg, the resulting diploid zygote, because again, two haploid cells coming together would double the chromosomes and make a diploid zygote. Then it divides through mitosis to form a new multicellular diploid sporophyte. In seed plants, they're unique because the sporophyte is the dominant part of the plant. It's what you see when you look at a seed plant. You're looking at a diploid plant that's a sporophyte. Another note I'd like to point out in this image here is that the green half, the bottom half, is the diploid half. So you see 2N in the bottom left. And so the only diploid components of a plant are going to be that fertilized egg that becomes the zygote and the sporophyte. All right. Another important difference is in the transition to seeds is actually spore production itself. And that leads to a defining feature of male and female, which is the difference in gamete sizes that are produced. Most, but not all, seedless plants are homosporous. This means that the spores that are generated from the sporophyte are of a single size. That one spore type develops into a gametophyte that produces eggs and sperm. Seed plants, by contrast, are heterosporous. So the sporophyte will either generate a larger, called megaspore, or a smaller spore we call a microspore. The megaspores develop into female gametophytes, which make the female gametes, these are the eggs. And the egg is built to be stationary and larger gametes. This allows them to specialize in developing and gathering resources needed for fertilization. The microspores develop into male gametophytes that produce sperm. You can see them beside the you can see them beside the megaspores there. These male gametes that are generated from the microgametophyte don't need all of the resources and don't need to be as big as the mega gametophytes gametes need to be, because their job is just getting the genes to the egg. In seed plants, both male and female gametophytes remain dependent on the sporophyte tissue. So they remain with the sporophyte for both water and nutrient supply, and which is why they can be reduced to these microscopic structures inside the sporophyte. This figure lists plant groups based on their adaptations. We covered this in more detail in our last presentation, but mosses and other non-vascular plants, ferns and other seedless vascular plants, and lastly the seed plants, all of which are vascular, are highlighted here. Highlighted within the red box are the seed plants. You can see that the sporophyte, let me get out my highlighter here, the sporophyte is considered to be the dominant structure of the seed plants. So when you look at these plants, what that means is you're looking at basically the entire plant that you're able to visualize is going to be that diploid sporophyte. The gametophyte portion is very small and living within the sporophyte dominant plant. And now that we can see the life cycle, how the alternation of generations has evolved in seed plants, we can discuss how that allowed seed plants to triumph over the water requirement of fertilization and reproduction and brought about a variety of other advantages as well. Really quickly, let's define two structures that give seed plants water independent fertilization. A seed is the part of a plant that contains the embryo and a protective coat. It's the beginning of the sporophyte, and the seed coat shields it from desiccation. A pollen grain is also a protective structure for the male gamete. The pollen grain is the pollen grains are the cells that eventually produce sperm, and it is made with a tough wall that allows transport without drying out the internal cells. These two durable structures work together to free the gametes from water-dependent fertilization. Now that we understand how pollen and seeds contribute to the success of gymnosperms and angiosperms, these spermatophytes as we call them, we need to become familiar with the actual parts of a flower so we can follow the reproductive process. The large labeled diagram is what is referred to as a perfect flower. This is one that contains both male and female reproductive organs. We're using a perfect flower in this slide because it's got everything we want to cover all in one flower. Let's go through the structure in the order they appear on the slide, starting from the outwardly parts of the flower and moving inward. The petals are the colorful parts most people notice first. Collectively, they are called the corolla, and they are modified leaves that surround and protect the reproductive organs. Their bright colors, scents, and sometimes ultraviolet patterns serve as advertisements to attract pollinators such as bees, butterflies, birds, even beetles. Just outside the petal, and there's other pollinators, of course, as well, but just outside the petals are what we call the sepals. Collectively, they form the calyx. So we have the corolla and the calyx. And they are also modified leaves. While the flower is still a bud, the sepals enclose and shield the more delicate petals and reproductive parts inside from damage, desiccation, and even herbivores. Then we have what's called the perianth, and this is the corolla and calyx combined, so all of the sepals and all the petals together. So listed as sets, we have the corolla or the petals, the calyx or the sepals, and the perianth, which includes both. The penducle is the stalk that attaches the entire flower to the rest of the plant. It provides both mechanical support and a pathway for vascular tissue to supply the flower with water and nutrients. The receptacle is the thickened part. It's right here. This is the thickened part of the stem at the top of the peduncle. And this is the base upon which all of the floral organs are attached. The receptacle may become enlarged and contribute to the formation of fruit later on. So we have the peduncle, which is the stem part, and the receptacle, which is where the flower parts are attached. This brings us to our first assessment question. These are just to assess how well you're covering the material as we go along. This is checkpoint 1. What part of the flower is made up of both the sepal and petal? Now we reach the reproductive organs themselves, beginning with the female part. At bottom left is the gynoecium, also called the carpel. It's the female sex organ of the flower. It consists of 3 regions. At the top is the stigma, the sticky or feathery surface where pollen grains land and are collected. Below the stigma is the style. This is a slender channel that connects the stigma to the ovary and through which the pollen tube will later grow. At the base is the ovary, the swollen structure that houses the ovules. Inside each ovule is the female gametophyte and the egg cell waiting to be fertilized. After fertilization, the ovary wall will thicken and mature into the fruit. The male part is the androecium, most commonly referred to as simply the stamen, shown here at the bottom right side. Each stamen consists of a thin stalk called the filament that supports the anther at the top. The anther is the sac-like structure where pollen grains, which are the female gametophytes, are produced inside the microsporangia. A whorl is a general term used to describe a circular ring of similar floral parts. So this isn't as specific as a corolla or a calyx. It's just saying these are a circular ring of similar floral parts. So it could be the sepals or the petals, the stamens or carpels, attached at the same level around a central axis, so like the receptacle of the flower. A flower is described as complete when all 4 whorls are present. Those 4 whorls are the calyx formed by the sepals, the corolla formed by the petals, the androecium formed by the stamen, and the gynoecium formed by the carpels. If every one of these 4 major sets of organs, or if even one of them is missing from the flower, then the flower is classified as incomplete. This distinction is important because it explains much of the structural diversity we observe across flowering plants and helps us understand how different species have adapted their reproductive structures to specific pollination strategies. Not every flower looks like the perfect flower we examined earlier. Flowers can be classified by whether they contain both sex organs or only one. Perfect flowers have both the carpel and the stamen on the same flower, so they are technically bisexual flowers. Imperfect flowers have either male or female parts, but not both. If a plant produces separate male and female flowers on the same individual, we call the plant monoecious. If the male and female flowers occur on entirely different plants, the species is called dioecious. Many plants also produce inflorescences. These are clusters of many small flowers arranged together on a single stem. The photograph on this slide shows imperfect beech inflorescences. with the female cluster at the upper left and the male cluster at the lower right. This arrangement is common in wind-pollinated trees and can help us understand why some species separate the sexes spatially. Checkpoint 2. Theobroma cacao produces clusters of flowers that grow directly from the trunk of the tree. They must be pollinated by tiny midges called chocolate midges or cacao gnats. What kind of flowering arrangement is this? Incomplete flowers can be further described according to which sex organs they possess. Staminate flowers contain only an androecium, while carpellate flowers contain only the gynoecium. The figure on this slide uses the corn plant to show this distinction in a familiar crop species. The staminate flowers are clustered together in the tassel at the tip of the stem and are responsible for producing the pollen grains. You can see them here on the left side. The carpellate flowers are clustered in the immature ears, and each long strand of silk extending from the ear is actually an elongated stigma waiting to receive pollen. After fertilization, the corn kernels that develop on the ear are the resulting seeds. The diagram also includes the lower stem and root structures, but we don't need to get into that right now. But this would be an example of a monoecious plant. It's a type of grass. It has these fibrous root structures here, which are a great indication of it. And we'll discuss more about that later, though. One final structural detail that is especially useful for plant identification and fruit development is the position of the ovary relative to the other flower parts. The ovary can be superior, meaning it sits above the attachment point of the sepals and petals and stamens. Alternatively, the ovary can be inferior, meaning it sits below the attachment point of the other flower parts. This difference is very important because it influences how the fruit will form and expand after fertilization. The figure on the slide demonstrates both conditions clearly. Part A shows the lily as an example of a superior flower. You can see that that this is found up the ovary above the where the attachment point is for the rest of the flower parts. Part B shows the fuchsia as an example, which has an inferior a flower, in which the ovary is located beneath the other parts of the flower. Notice how the relative position affects the overall appearance of the flower, and this will later affect the way the mature fruit attaches to the plant as well. This figure takes our perfect flower and divides the process of gamete formation and fertilization based on the female part of the plant or the male part. In the upper right corner is the life cycle absent of the structures to show the steps involved in the heterosporous plants. Remember that almost the entire plant is the sporophyte. Shown on the left side is the female portion called the carpel or gynoecium, containing the ovaries, which contain the ovules, and inside each ovule is the megasporangium and the female gametes. The megasporocytes are responsible for forming the haploid megaspores by going through meiosis. Since it is the female, these spores would be called the megaspores. Oh, let me back up here. These megaspores then germinate into the gametophyte. Again, in this case, we'd call it specifically the megagametophyte. Another name for which is the embryo sac. The megagametophyte is responsible for the production of haploid female gametes, which we call eggs. Similarly, if we start at the top with the male stamens, they go through the same steps. The microsporangium is found within the anther of the stamen, and the microsporocytes form the haploid microspores through meiosis. The microspores germinate into the microgametophyte, which produces the gametes, aka the pollen, and this pollen, if successful, will go on to fertilize the eggs. We are now ready to look at the ultimate goal of the flower, and that is fertilization, which occurs after a pollen grain lands on the stigma. At the bottom of the image we've been using to review the sex cycle, you can see the fertilization stage highlighted in that gray box. You'll see that from the male side, that's the right side, the pollen grain morphs into an unusual shape, forming what is called a pollen tube. The pollen tube is the vehicle that the pollen uses to deliver the gametes within the ovary. With the flower parts clear, and we've talked a little bit about the cycle, we can now look at what happens after pollination. In angiosperms, fertilization is unique because it involves two separate fusion events inside the ovule. This is called double fertilization. The figure shown here picks up in our discussion of when the pollen grain lands on the stigma and the vehicle to deliver the sperm nuclei, called the pollen tube, grows down the style or develops down the style. And so the nucleus of one sperm from the pollen tube then reaches and combines with an egg cell, and that forms the diploid zygote. That zygote will divide by mitosis to become the embryo, which again is that the young sporophyte inside the future seed. But we said this is called double fertilization. Okay, so what this means is there's a second sperm nucleus, and that fuses with what we call two polar nuclei that are inside the central cell of the female gametophyte. This is very unusual, and there's a purpose for this. So this second sperm nucleus, again, it fuses with two nuclei of the other plant, of the egg cell, and it creates a sort of strange triploid cell because there's two polar nuclei from the female gametophyte and then one nucleus from the sperm, so that's three, triploidy cell. And this triploidy cell divides to form the endosperm. And the endosperm is a nutritive tissue that's packed with starches and proteins and oils, and that's going to feed and help develop that young sporophyte within the seed, that developing embryo during germination. Double fertilization, therefore, produces both the next generation of the plant and a dedicated food supply for that generation, giving angiosperm seeds a tremendous head start compared with gymnosperm seeds. So next, we'll just watch a video of this process that goes into more depth on double fertilization. You don't need to memorize anything in greater detail than I just covered here, but it's going to help improve your understanding of the steps that lead to the generation of fruits. Here is that short video on double fertilization in flowering plants, aka the angiosperms. And it's zooming in right now onto the androecium, which are the little anthers that surround the gynoecium, which is the stigma and style of the female reproductive organ. There's going to be some changes that need to occur first with the female reproductive portion. So here's one megaspore, and it actually goes through a series of divisions. It goes through meiosis and creates the spores, the megaspores. Interestingly enough, three of them degenerate and only one of them survives, and it expands and takes up the whole space here. Now, the nuclei are going to copy. This is not a whole new cell. This is just the DNA that's being replicated and copied into new nuclei. So they're all in the same cytoplasm, and they're going to move to their specific loci of this cell before any cell walls form around some of them. And now we have what we call the embryo sac. Cell walls begin to form around some of the nuclei here. Okay, now 3 more cells form here above the micropyle. The one that's kind of a pinkish red, that's going to be the egg cell that becomes the zygote later. Now, 2 cells in the middle don't form a cell wall, and that's going to be important for the second fertilization that takes place. Here we go. It's going to zoom in now to the anther and highlight a pollen grain. The pollen grain contains 2 cells. One cell called the tube cell, and then a sperm cell, also called the generative cell. Here it lands at the stigma of the gynoecium. And the pollen grain cell is going to form the pollen tube that the germ cell can travel, or generative cell can travel along. It's going to now reach the micropyle, and then mitosis allows that germ cell to duplicate. The sperm cells get released. One of them immediately fertilizes the egg cell. Now it's a diploid zygote. Now the other sperm cell is going to fuse with those 2 central nuclei, becoming the triploid cell. And that is what goes on to make the endosperm, which is a nutritive source for the seed. And that only occurs in angiosperms, because angiosperms make fruit, and that is going to go on to become the part of the plant that gives it, the seed, the name of fruit. Hopefully that video was a great visualization of what double fertilization is. To summarize the key points of this process, 2 sperm cells from a single pollen grain fertilize 2 different cells within the female gametophyte. One sperm fertilizes the egg to form the diploid zygote. That becomes the embryo. The other sperm fertilizes the central cell to form the triploid endosperm that provides nutrition. After these fusions occur, no additional sperm can enter the ovule. The fertilized ovule will mature into the seed, while the surrounding ovary tissues will develop into the fruit. That often encloses and protects the seed. This coordinated process is one of the major reasons angiosperms have become the dominant group of plants on Earth. Checkpoint question 3. What is the function of the unique other sperm cell in angiosperms, not seen in gymnosperms, in the fertilization process? We've shown this table before in prior presentations. It's a table of adaptations from land plants. The bottom 3 are strictly found in seed plants, with the development of flowers and fruits only found in angiosperms. Seeds are protective structures surrounding plant embryos. They contain storage food and a tough coat that enables the embryo to survive dispersal and remain dormant until conditions are favorable. Flowers are specialized reproductive structures that bring male and female gametophytes together in one place and facilitate precise pollination. Fruits are thickened tissues that provide the plant embryo and aids dispersal, usually by attracting animals or using wind and water. Together, these innovations explain why seed plants dominate our terrestrial landscapes today. And so they're going to be a major focus of this presentation. We'll first begin with the gymnosperms that share the seed structure adaptation with angiosperms, though. Although angiosperms are the primary focus of this lecture, we need to understand how sexual reproduction occurs in the other major group of seed plants, the gymnosperms, so that we can appreciate the unique features of flowering plants. Gymnosperms also follow the alternation of generations life cycle that we examined earlier in this presentation and in the previous lecture on seed plants. In conifers, such as pines, the green leafy part that we recognize as the tree is the sporophyte generation. The male and female gametophytes are held inside cones rather than flowers. Female cones are larger and usually found on the upper parts of the tree. This is by design. The smaller male cones are typically located in the lower regions. What this does is it makes an arrangement that reduces the chance of self-pollination because wind doesn't usually blow in a nearly straight upward direction. So for the pollen to reach the upper cones, it would have to fight against gravity. The wind-dispersed pollen from the male cones must travel through the air to reach the female cones since gymnosperms rely on wind for pollination. They produce large quantities of pollen to increase the probability that some of those pollen grains will land on the correct target. Now we can directly compare the reproductive strategies of angiosperms and gymnosperms in order to highlight what makes flowering plants distinctive. Gymnosperm reproduction differs from angiosperms in several important ways. In gymnosperms, the female gametophyte develops on exposed, what we call bracts. This is the little pieces of the cone of the female cone. Whereas in angiosperms, the female gametophyte is enclosed and protected inside an ovule that it is itself contained within the ovary. Double fertilization is completely absent in gymnosperms, which lack the ovary tissues that are derived from the secondary fertilization and lead to what we call the fruit. But it's the key event in angiosperms that produces both the embryo and the nutritive endosperm. Male and female gametophyte structures are located on separate cones in gymnosperms, but they occur together within the same flower in angiosperms quite often. Pollination in gymnosperms depend almost entirely on wind, with some key exceptions we discuss in the lecture on seed plants, with pollen being blown onto the female cones, while animal pollination is much more common and often quite more efficient in angiosperms. Finally, in primitive gymnosperms such as ginkgo and cycads, you'll recall those were the first gymnosperms in the fossil record. The sperm are still flagellated. However, these sperm are delivered to the female gametophyte while still enclosed inside the pollen grain, and they're released only for a short travel to unite with the eggs. These differences show how angiosperms evolved additional protections and efficiencies that contributed to their success. Once the zygote and endosperm have formed, the embryo begins to develop inside the seed. The first embryonic leaves to appear on that embryo are called cotyledons. The number of cotyledons becomes the basis for dividing angiosperms into two major groups. Monocots have one cotyledon. Eudicots, often called dicots, have two. These cotyledons serve as the initial food transfer organs, moving nutrients from the endosperm or even nutrients that are stored within these cotyledons themselves to the growing shoot and root. If you look, for example, at these, just look at the eudicots here, or the monocots here. You look at the seeds, look how the majority of what you're seeing here, these giant portions, that's the cotyledon. Let me pull up my laser pointer. That's the cotyledon in the seed. So there's two of them here in the eudicot, and they're very large, much larger than just a regular leaf. And that's because they're often storing nutrients in there, starches, proteins, things like that. Look at the seed of this monocot. It's huge, and it's so large because of that cotyledon, which stores a lot of nutrition there. So they can be a source of that nutrition, as well as they can just be a mode of transfer of nutrition from the endosperm to the rest of the growing shoot and root. So If you look at any plant, there'll be a shoot that's coming out one end and a root that's coming out the other. All right, I just want to say too, examples of monocots are going to include wheat, corn, rice, onions, and palms. We discovered many of them in the last presentation. Examples of eudicots include peaches, sunflowers, tomatoes, beans, cacti. This diagram here illustrates how the single cotyledon in monocots versus the pair in eudicots affects the overall early growth pattern after germination, which we'll cover in more detail when we go through both of these groups. Within the seeds of both monocots and dicots, we can identify the embryonic structures that will give rise to the major parts of the new plant. The radicle is the embryonic root that grows downward and develops into the root system of the seedlings. The plumule is the embryonic shoot that will develop into the leaves and stems. The hypocotyl, hypo means below and cotyl for cotyledons, so it lies below or between the radicle and the cotyledons, the hypocotyl. It grows upward and forms the main stem. In monocots, after germination, the primary root that rises from the radicle, it actually soon dies. And what emerges is what we call adventitious roots. These actually grow directly from the stem itself, and they take over. And because they grow straight from the stem, they grow several roots out instead of a major root. And that becomes a fibrous root system. The diagram on this slide shows the internal organization of both types of seeds, so please review it to see exactly how these embryonic regions are positioned relative to one another and how they will contribute to the mature plant body. Checkpoint question 4. Where is the hypocotyl located in relation to the cotyledons? Monocots are one of the two major derived groups of angiosperms and can be recognized by a consistent set of structural features. Their leaf veins run parallel to the length of the leaf rather than forming a network. The stem tissue is almost always herbaceous. That means it's kind of green and soft, and it rarely develops into true, hard, a woody secondary growth. Monocots produce adventitious roots, which means that they emerge directly from stems or other non-root systems. And they form a dense fibrous root system with no single dominant taproot. Finally, their flower parts, such as the number of petals and sepals, typically occurs in multiples of 3. These traits are easy to spot in the field. All grasses, including sugarcane, cereals, rice, corn, and wheat, are monocots, and they'll share many of these traits. Except for the flower petals, that does seem to vary quite often, but the other 3 regarding the roots, leaves, seeds, they're all going to follow those traits. So are orchids, palms, pineapples, and bananas. I recommend you even take some time just looking at those examples and see if you can spot those different characteristics. And before you know it, any plant you could be easily able to distinguish whether or not it's a monocot. This brings us to the angiosperm dicots, or more accurately called the eudicots, meaning true dicots. They make up the other major derived group and show the contrasting set of traits. Their leaf veins, if you look right down here, the leaf of the eudicot, you'll see that it actually forms a net-like or branching pattern where the veins come off to the sides here instead of running parallel with the whole length of the leaf. Woody tissue with secondary growth is quite common in this group, and this allows many dicots to become large trees and shrubs. They usually develop a taproot with several lateral roots branching from it. So they don't have the adventitious roots that you see in this example here at this bulb with the very numerous roots that grow around the base of the stem. Their flower parts, they grow in typically multiples of 4 or 5, but again, I want to just impress upon you that the number of flower sepals or petals really varies a lot between these two groups, especially with modern breeding of many of our dicots. It's not exactly the most reliable way to identify a dicot or a monocot. But examples surround us every day. So, sunflowers, beans, most of the culinary fruits that we eat, such as peaches, grapes, and oranges, and vegetables, such as broccoli and potatoes, these all are dicots. And if you take a little bit of time looking at some examples of their leaves and their seeds and their root systems, I'm sure you're going to get the hang of telling which plants are eudicots or not. Having examined the overall characteristics of monocots and eudicots, we can now look more closely at one of the most important differences between them, and that's their root systems. So in dicots, the primary root develops directly from the radicle of the embryo. Secondary lateral roots then rise from the primary root. So if you ever were to like look at a carrot in the store, you might see some lateral roots that are secondary that come off of this primary root. And a carrot is like an extreme example of the taproot, right? This has been bred to create a overly whelming dominant taproot, but you can still see those little lateral roots that do form. Those lateral roots tend to be a little bit bigger, of course, in other plants such as trees, but nonetheless, that has those two-part components to its roots. Those secondary lateral roots, as I said, they do arise still coming from the primary root. So they're not growing off from just the stem at the very top like you'd find in monocots. And this sort of system allows that plant to reach deeper for sources of water with that taproot, and it provides an excellent anchorage. Um, with that said, you know, deep roots, in general, that means they're able to acquire water that's deeper into the soil that doesn't maybe get evaporated off like the really shallow layer of soil might. But some plants, in particular some dicots, have evolved very shallow roots. And the reason for that is cacti and succulents that grow in the desert. When it does rain, it doesn't rain enough to really get that deep soak into the soil. And so having those shallow roots does allow those sorts of plants to take advantage of the little spritzing of water that comes with the intermittent rain showers. In dicots, again, the primary root that originates from the radicle, it persists and becomes the main taproot. In monocots, after germination, the primary root soon dies off. Its job is complete in establishing the plant in the soil. And then adventitious roots emerge from stem cells rather than from root tissue or root cells. So the stem cells convert over into root cells. These adventitious roots give rise to the characteristic fibrous root systems we see in grasses and corn. If you live in Arizona and you walk around the neighborhood and you look at the palm trees, you can just see the shallow adventitious roots coming off of the stem of the palm. This dense, shallow network is excellent at preventing soil erosion, but it doesn't typically penetrate as deeply as taproots do. So this slide is just going to emphasize that key difference between the two of getting used to identifying these different features in the roots. It comes a long way in helping you identify what type of plant it is. And checkpoint 5 says blank have adventitious roots and blank veins in their leaves. Moving along, examining the parts of a flower is one of the best ways and very important ways for identifying what type of flowering plant you might have. In the next few slides, we'll look at some eudicot examples of flowers and the families they belong to. Accompanied with each plant family are images that show how they can be identified. Much how we learn that fungi are often identified by examining the fruiting bodies, So can every flowering plant family. And it typically includes counting the number of each body part within the flower. So one of the most important families we'll start off with here in the eudicots is the Brassicaceae, also known as the mustard or crucifer family. It contains more than 4,000 species, and remarkably, every single one of them is edible, although they might not be all tasty, so to speak. The most famous member is Brassica oleracea. This single species has been artificially selected by humans and bred in order to accentuate different parts of the plant. So, if you can imagine cabbage or kale, they've been bred to accentuate the leafy parts of the plant, or cauliflower and broccoli have been bred to accentuate the flowering parts of the plant, along with collards. The floral diagram on this slide shows the classic crucifer pattern and where it gets its name. Four petals are, and sepals that are arranged in the shape of a cross. This is why the family was historically called Cruciferae. However, it has been renamed Brassicaceae um, to be after its main representative member that humans use, the brassica. And this sort of plant or sepal and petal arrangement also comes with six stamens. And if you look at the image on the right, where each one of the plant parts are separated, the stamens, you'll note that actually four of them are tall and there's two of them that are short. And this sort of radial symmetry and fixed number of parts, it's typical of many eudicot families, and it helps confirm classification. The sunflower family, Asteraceae, is possibly the largest of flowering plants with approximately 32,000 species. You'll find many of these plants in the wild as well as in people's gardens. And when you know how their unique flower type, you should be easily able to identify it within this plant family. What looks like a large single flower on a sunflower or daisy is actually a composite head or capitulum. This is made of many much smaller individual flowers packed tightly together. Some of those tiny flowers are ray flowers with long petals that form the quote unquote petals we see around the edge, while the center is packed with disc flowers that produce the seeds. This functions all together like one giant flower, attracting pollinators more efficiently. The photographs on the slide are typical composite heads from different members of the family, so you can see the pattern clearly and across the whole family of Asteraceae. All right, now we'll move into Rosaceae, or the rose family. Rosaceae contains about 5,000 species and includes many of the fruits and ornamental plants we value most. Edible members include apples, pears, raspberries, strawberries, peaches, apricots, plums, cherries, and almonds. The flowers of this family are radially symmetrical with five sepals, usually five or multiple of five petals, and many stamens, as you can see in this image here to the right. A key structural feature is the hypanthium, a cup-like structure formed by the fusion of the sepals, petals, and stamens at the base of the flower. And the hypanthium that later becomes part of the fruit itself in apples and related species. The image on this slide shows a longitudinal section of an apple, and it illustrates how the fused floral parts of the hypanthium at the base expand as the fruit develops and contributes to a major portion of, or the edible portion that we eat. This fusion explains why the core of the apple sits inside what used to be the ovary, while the fleshy part develops partly from the hypanthium. So all of this fleshy section here kind of makes a C around the ovary core is from the hypanthium. After fertilization has taken place, the ovary of a flower typically transforms into a fruit. We often associate fruits as sweet parts from a plant, but as we should know by now, fruits are defined by their origin and development from the flowers of angiosperms. From a botanical perspective, the term fruit has a precise meaning. A fruit is a ripened ovary. This definition is based on developmental origin from the gynoecium. Fertilized flowers usually develop into fruits, while unfertilized flowers typically don't. We distinguish true fruits from accessory fruits by the origin of their tissue. So where they are derived from the flower. True fruits develop strictly from the ovary wall. We saw how apples and pears from the Rosaceae family have a large portion of their flesh that develops from the hypanthium. So they would be considered an accessory fruit because this doesn't come from the ovary itself. In every case, fruits protect the seeds and developing embryo by enclosing them completely. The fruit wall acts as a physical barrier against drying out mechanical damage and many herbivores until the seeds are mature enough for dispersal. Because fruits develop through different pathways and serve different dispersal strategies, biologists categorize them by origin and final structure. After double fertilization has taken place inside the ovule, the next major step is the development of the fruit. The fruit forms from two parts of the flower that we examined earlier: the developed ovule, which becomes the seed or seeds, and the thickened ovary walls, which expand and mature into the fruit structure itself. This process begins right after fertilization when hormones trigger rapid cell division and expansion in the ovary wall. Many foods that we casually call vegetables are actually fruits in the strict botanical sense because they develop from the mature ovary. Tomatoes, for example, are berries that develop from a single ovary, and beans are dry fruits that split open to release their seeds. Fruits themselves fall into two broad categories based on the nature of the ovary wall and maturity. Fleshy fruits have a soft, juicy layer called the pericarp. This attracts animals. Examples include berries such as blueberries, stone fruits such as peaches and grapes, um, and also grapes, sorry. And dry fruits have a hardened pericarp and are often adapted for other dispersal methods. Examples include the grains of rice and wheat, as well as nuts. Nuts are actually um, um, part of a fruit as well. Carpels are the individual units of the gynoecium. To make sense of fruit diversity, we classify fruits by how many carpels or flowers contribute to the final structure. Simple fruits develop from a single carpel or from carpels that are fused within one ovary. Because only one ovary is involved, all seeds inside trace back to the same flower. Nuts and beans are a great illustration of this type. Aggregate fruits develop when multiple carpels within the same single flower each form a small fruit, and then those small fruits fuse together during maturation. So if you look at this image right here of the raspberry flower, it shows the stigma and the style are each of these individual ones here, and they each have their own individual carpels that are going to develop into their own fruits, and then they fuse together. And you see how it's shaped like a cone here. That is what creates this aggregate fruit. So raspberries show this pattern quite clearly, with each bump as a separate fruitlet from its own carpel. Multiple fruits form when an entire inflorescence or many separate flowers, that's the key part here, grows together. Each flower produces a small fruit, and these fuse together into one large structure. Pineapples are a great example of this. What we eat, there are combined matured ovaries of dozens of separate tiny flowers that were clustered on one stem. And finally, accessory fruits originate from tissue other than the ovary. The main fleshy part comes from the receptacle or hypanthium, while the true small fruits are embedded within or on the surface. Okay, so a great example here is of a strawberry where the receptacle forms that fruit in the accessory fruit. So strawberries develop from the receptacle, and there you have it. Whereas apples and pears develop largely from the hypanthium, and so this is another example of an accessory fruit. Oh, and by the way, it's called an accessory fruit because these are accessory tissues. They're not part of the main ovary. Checkpoint question 4. What distinguishes accessory fruits from other fruit classifications? Let's continue our discussion of the fruit layers. The fruit wall that develops from the ovary after fertilization, again, it's called the pericarp. And this pericarp, it's actually organized into three distinct layers. Starting from the outside in, we'd have the exocarp. And then the middle portion is called the mesocarp. You can follow along in this image of a peach here. And then we have the inner portion called the endocarp. And these three distinct layers determine the fruit texture and how it protects the seeds. In many fruits, the mesocarp thickens and becomes that juicy, edible portion that attracts dispersers. In other fruits, like almonds, the endocarp, that's the inner portion of the pericarp, it hardens into the shell we crack to reach the seed. So this actual seed is inside of that. So if you look at this image here, this peach, this stony, really hard component, if you crack it open, then you release the seed. So this isn't the seed layer on the outside. That's the most inner portion of the pericarp. Okay, and I just want to say that over time, in many fruits, these three layers, they can expand. One of these layers can expand, or some of these layers can fuse together so completely that the boundaries become difficult to see at maturity. And this fusion is a normal part of maturation after double fertilization. And the image to the left is a peach. The exocarp is the thin outer skin. The mesocarp is that thick, juicy flesh. And the endocarp is that hard stone that encloses the seed. And the pair to the right, the layers are similar, but part of the fleshy tissue comes from, again, the hypanthium that was incorporated during development. Fruits can be grouped as dry or fleshy. We've discussed this before, depending on whether the pericarp stays hard or becomes soft. And they are also grouped as dehiscent or indehiscent. And that depends on whether they open to release the seeds on their own. So does the pericarp open or not determines if it's dehiscent or indehiscent. So dehiscent fruits are going to readily release the seeds, whereas indehiscent fruits, they're going to require some sort of decay in order, or some help in order to release their seeds. Mature fruits must move seeds away from the parent plant to reduce competition around that parent plant. and colonize a new area. And here are three common dispersal methods. Dandelion seeds are attached to lightweight feathery structures that catch the wind and carry them long distances. Coconut fruits have a thick fibrous husk with air pockets that allow them to float on ocean currents and reach distant islands. Acorns are collected and cached by animals such as squirrels. When the animal fails to retrieve this nut, then it might germinate and form into another full tree. And each one of these are examples of dicots, and we're looking at the varying fruiting structures. And so each of these strategies just depends on how the pericarp layers are formed. Dehiscent fruits, again, they open naturally when they reach maturity. It's triggered by drying out and shrinking of the outer regions. And when the region dries out and it shrinks, that creates a lot of tension that builds up in the exocarp or the outer layers of the pericarp. As a result, the seeds are released directly into the environment where wind, animals, or gravity can carry them away. And this mechanism is quite efficient for plants that produce many small seeds and don't rely on animals to eat the fruit. These include plants like beans, peas, even poppy capsules are types of dehiscent fruits that split along built-in seams that help free the seeds up. Indehiscent fruits remain closed at maturity. Their seeds stay protected inside the fruit until external forces act on them. These fruits can be fleshy like strawberries and raspberries or dry like acorns and nuts. Seeds are dispersed when animals eat the fruit and pass the seeds and droppings, when the fruit decays, or when animals carry and drop them. This strategy maintains genetic diversity by spreading seeds over wider areas so that they don't stay so close to each other and have more interbreeding with closely related plants. And this is especially common in plants that invest in fewer but better protected seeds. Not all plant reproduction requires pollination, seeds, or flowers. Asexual reproduction allows a plant to reproduce genetically identical offspring without mixing gametes. This method is useful in stable environments because the new plants are already well adapted to the same conditions. To understand how it works, we need to examine structures outside the flower, such as shoots and roots. Stems are part of the shoot system, the upward growing part of the plant. They are the part of a shoot of the shoot system that supports leaves, flowers, and buds, and sometimes store food. Stems connect roots to leaves and transport water, minerals, and sugars throughout the plant. The stem is organized with nodes where leaves and buds are attached, and internodes between those attachment points. The stalks that connect a leaf to the stem is called the petiole. Auxiliary buds are located in the axil, that's between the leaf base and the stem, and can grow into new branches or flowers. The tip of the stem is the shoot apex. You can see it here, sometimes having an apical bud that drives upward growth. These features allow stems to produce new plants through asexual means, as we'll see next. But first, let's do a checkpoint question. Checkpoint 6. Where are auxiliary buds typically found on a plant stem? Stems contain two specialized conducting tissues that move resources through the entire plant body. Xylem stores and transports water along with dissolved minerals from the roots up to the leaves and growing tips. Its cells have walls that are strengthened with lignin, so the tissue can withstand the tension created when water is pulled upward within them. Phloem transports sugars, proteins, and hormones produced during photosynthesis from the leaves to all other parts of the plant, including to the roots, other stems, and developing fruits. These two tissues run side by side and work together to support both daily function and the growth that leads to reproduction. I've always remembered the difference between the two with the phrase xylem goes up and phloem flows down. Roots are organized into two main systems that differ in structure and function. A taproot features one main root that grows straight down with smaller lateral roots branching from it. We saw this was common in monocots, like the dandelion, which has a taproot that can regenerate an entire plant from a small remaining piece. These taproots allow the plant to reach deep water sources and provides a strong anchorage in the soil. A fibrous root system consists of many roots of roughly equal size that spread out close to the soil surface in a dense network from the stem. This design is excellent at holding soil in place and quickly absorbing water from the upper layers after rain. Monocots, like lawn grasses, wheat, rice, and corn, all have fibrous roots. The type of root system a plant has directly affects how it obtains resources and how well it survives a disturbance to the soil. Some plants have a combination of the two. Deep roots reach deep soil. Shallow soil tends to dry out quickly, but we've discussed earlier that that's not always the case when it comes to roots such as cacti and succulents that have shallow roots to capture light rain. A root development starts at germination when the radicle of the embryo grows downward to form the first root. At the very tip is called the root cap. As you can imagine, as it's burrowing into the ground, it needs a protective layer of cells that is continually worn away and replaced as the root pushes through the soil particles, and that's called the root cap. Immediately behind the cap, there are three functional zones. The zone of cell division contains actively dividing, we call them meristem cells that produce new cells. Then we have the zone of elongation. This is where new cells rapidly increase in length, pushing the root tip forward. Again, this is just right behind that root cap, very close. And then we have the zone of maturation. This is where cells complete their differentiation into mature types. The outer surface is the epidermis, which produces root hairs that are extensions of single epidermal cells. These root hairs dramatically increase the surface area available for absorbing water and mineral ions from the soil. Plants can also reproduce without the flowers, pollination, or seeds, as we said, and many plants have natural asexual reproduction strategies, which inevitably requires either generating the new roots or shoots that we went over. Some species produce buds on the surface of underground or above ground stems, such as ginger, onion, gladioli, or a dahlia. And you can see from this image right here, you can see the buds forming underground. And this comes from the meristem, the stem tissue, that's capable of differentiating into root cells to grow into a completely new plant. When conditions are right, cells in the bud differentiate to form roots and shoots, creating a genetically identical offspring. Other plants use specialized horizontal stems called runners or stolons that grow along the soil surface. At each node that touches moist soil, stem cells differentiate into adventitious roots, and they form a new shoot from there as well. Sweet potatoes and strawberries rely on this method because the new plant develops directly from the parent stem tissue. It is genetically identical and quickly establishes itself in the same environment. Finally, many plants can be propagated from simple stem cuttings, as long as the stem cell retains the ability to differentiate into root cells. A cut piece placed in suitable conditions will develop adventitious roots and become a new independent plant. The image shows how applying different growth hormones encourages this differentiation from stem to root cells and the speed and success of root formation and cuttings. We'll end here on our presentation of plant reproduction and structure, and I hope you found it interesting, and please join me for the next one.